The overall goal of this procedure is to characterize the proteomic composition of functionally distinct chromatin domains in order to understand how the different elements synergize to determine the transcriptional state. This is accomplished by first preparing nuclei from cultured cells and digesting chromatin into fragments of optimal length through enzymatic or mechanical means. The second step is to purify a distinct functional domain from bulk chromatin by immunoprecipitation using bait antibodies against either a histone post-translational modification or a protein known to specifically mark the region of interest. Next, the immunopurified chromatin proteins are separated by SDS-PAGE and in-gel digested prior to mass spectrometry. The following step is the liquid chromatography mass spectrometry analysis on a high-resolution instrument. Ultimately, mass spectrometry raw data are analyzed by ad hoc software, followed by manual inspection of the spectra to identify and quantify histone modifications and co-enriched proteins, based respectively on either the calculation of the relative abundance in immunoprecipitated material relative to input, or on the Silac ratio in competition experiments using soluble peptide as competitor to the bait.
Through ChroP, we can gain insight into the interplays between histone modification pattern and variants enriched at specific chromatin regions and the nuclear interactor recruited thereby, which